Now, just before we start talking about the mechanism of respiration and some of the reactions that are involved and some of the enzymes, it might be worth you just pausing the video for a short time and having a think through these 5 different questions about respiration. So this would really just be a little bit of revision, just to kind of look at where your current understanding lies. So if you want to pause the video, I will then go through the answers on the next slide. Okay, now if you've had a go at those questions, the first one asked for the balanced chemical formula for aerobic respiration. So we know in aerobic respiration, oxygen is going to be required as a starting material. Now, not just oxygen, but also the monosaccharide that we call glucose. And we know glucose is a hexose sugar because it has 6 carbons in it. So glucose is C6H12O6. We know molecular oxygen is O2, and there's going to be 6 molecules of oxygen. We know the waste products of aerobic respiration are water and also carbon dioxide. So for each glucose molecule, we will use up 6 oxygen molecules, and we will release and then excrete 6 carbon dioxide molecules. Now, the whole point of aerobic respiration is that energy gets released in the gradual breakdown of glucose, and it gets used and converted into what we call ATP. Now, ATP is a universal energy currency of the cell. So the ATP molecule can carry small packets of energy inside the cell to where it's required. Now, aerobic respiration occurs inside all different types of cells, eukaryotic cells that contain mitochondria, in some bacterial cells, and also obviously some yeast cells that also contain mitochondria. Now, aerobic respiration then, it's basically the complete breakdown of a molecule of glucose inside cells in the presence of oxygen to yield energy in the form of ATP, the universal energy currency. Now, we also need to know a little bit about anaerobic respiration. So again, that's the breakdown of glucose inside cells, but this time in the absence of any oxygen. So cells can often break down glucose to release some of the energy in glucose, even in the absence of oxygen, and that's going to yield a small amount of ATP in this case. So the amount of ATP that's released through anaerobic respiration is smaller than we would get in the presence of oxygen through aerobic respiration. Now, alcoholic fermentation is one form of anaerobic respiration, so we can put that at the top. Alcoholic fermentation, and we'll talk about that chemical reaction very shortly. And the other type of anaerobic respiration is lactic acid fermentation, or lactate production in, for example, muscle cells or even red blood cells inside our own body. Now, the universal energy currency in all organisms, whether they be bacteria, which are prokaryotic cells that lack any organelles, or fungi, such as the unicellular yeast, which is a type of fungus, or animal cells, which we know are eukaryotic, plant cells, and even protoctista, things like amoeba, tista. So it doesn't really matter what kingdom the cells belong to, what organisms they belong to. They will all make ATP. Now, why is ATP needed as a universal energy currency inside cells? Well, many processes inside cells, such as the building of larger molecules like protein synthesis from amino acid subunits, requires energy. And we call them anabolic reactions, where more complex products are being made. We know things like carrier proteins in phospholipid bilayers quite often require the energy released from ATP. And that changes the shape of the carrier protein, and it allows things like ions or certain polar molecules to be pumped across a membrane against a concentration gradient, which is why it's active. Processes like bulk transport, endo or exocytosis also require the energy released from ATP. Other processes like DNA replication and enzyme catalytic function will also require quite often the energy from ATP. So we could actually also add enzyme reactions to that list. Now, if we start by looking at the structure of ATP, we know it's a nucleotide, and it's actually an RNA nucleotide. But the main difference between ATP and the RNA nucleotides that are used to make messenger RNA and transfer RNA is that there's 3 phosphate groups all linked together, connected to the ribose sugar. Now, an RNA nucleotide used to make messenger RNA and transfer RNA will have a single phosphate group connected to the green ribose pentose sugar. Now, in the exam, you really need to know the cartoon version of ATP. So we can see a nucleotide, it's made of 3 components. Adenine is the nitrogenous base. So it's a nitrogenous organic base, and it's always adenine in ATP. Now, nitrogenous just means that it contains nitrogen. Organic means it contains carbon and hydrogen. So we know this is a double ring structure, so we know it must be a purine nitrogenous base. Now, the adenine is connected to the ribose sugar, and we know that's a pentose. So that's why it's an RNA nucleotide. Now, the adenine and the ribose together is called adenosine. So that's why we call ATP adenosine triphosphate, because the adenine nitrogenous base linked to the ribose is the adenosine, and the adenosine then has 3 phosphates added to it, so we call it adenosine triphosphate. And we know the word tri means 3. So underneath at the bottom of the slide, we can see the molecular structure of ATP. And we can see the nitrogen there in the adenine base. We can see the ribose sugar, which will have 5 carbons. So we can go 1, 2, 3, 4, and number 5 carbon here at the top. And then we can see 3 phosphate groups. So I'll just circle the phosphate groups in red. So the first one is here. The second phosphate group is here. And the third phosphate group is here at the end. Now, if we look at the cartoon version at the top, we've got 2 bonds here that link the 2 terminal phosphate groups. And these are what we call very high energy bonds, phosphoanhydride bonds. So we're going to put here high energy bonds. And there's actually 2 of them. Now, when these high energy bonds get hydrolyzed, it releases the phosphate group that's attached at the end of the molecule. And that will also release some energy that gets used in different processes inside the cell. Things like active transport, enzyme reactions, DNA replication, etc. So we can look at the formation of ATP, and we got the molecule at the top. So we can see the adenine, which is the nitrogenous base. It's a purine. We can see the ribose sugar that is connected to, and we've got the 3 phosphate groups. Now, we know the 2 phosphate high energy bonds here can release some energy when the phosphate groups are cleaved off through hydrolysis. And that will form ADP, where we have 2 phosphate groups, not 3, with the released inorganic phosphates and also obviously the energy. So ADP plus inorganic phosphates, when they're joined together, the enzyme that does that is called ATP synthase. Now that reaction going this way requires an input of 30.6 kilojoules per mole of energy to form the high energy bond that links the terminal inorganic phosphate with ADP. So we call that endergonic. And we know that's an anabolic reaction because it's making a more complex product, which is the ATP. Now, the reverse of that reaction can also occur, and that's where the ATP is hydrolyzed to ADP plus inorganic phosphate plus the release of energy. Now, that's exergonic because it releases energy, and the energy is 30.6 kilojoules per mole that gets released. Now, the enzyme that does this, or the catalytic activity, is called ATPase, and that will add the water to the high energy bond, releasing the inorganic phosphate. So we know that's a catabolic reaction. So quite often reactions are linked together. So we might have a carrier protein in a membrane, and it might have ATPase activity. So it might bind ATP, it might hydrolyze the terminal phosphate from the ATP. That releases some energy, and that is then utilized by the carrier protein to change shape and pump a certain ion or polar molecule across against the gradient. Now, we know glucose is broken down through a series of chemical reactions. There's not just one single oxidation reaction that breaks glucose down. It's broken down sequentially. So we're going to need this acronym OILRIG, and that stands for oxidation is loss and reduction is gain. And that refers to either electrons, or in biology we quite often talk about hydrogen, because we know hydrogen is made up of a proton and an electron. So OILRIG refers to the loss of an electron or hydrogen because that contains an electron, and reduction is the gain of an electron or the gain of hydrogen because we know it contains an electron. So if we have this intermediate here, A, and we know that is reduced because it has hydrogen attached to it. If that loses the hydrogen, it actually becomes oxidized. But quite often the oxidation reaction is actually linked to a reduction reaction. So if we have another substrate, in this case B, we're going to see that gains the hydrogen. So A, we can see, loses hydrogen on the left, and that is therefore oxidized. We can see B gains hydrogen, so that becomes reduced, and we call this a redox reaction. So A is oxidized because it loses hydrogen, B is reduced because it gains hydrogen. Now on the left hand side we can normally see oxidation is a catabolic reaction. Now is that likely to be exergonic or endergonic? Well, we know catabolic reactions where we have simpler products compared to the starting substrate, they are exergonic. So in this case oxidation is likely to release some energy. Now the opposite reaction is called reduction on the right hand side. We know that's anabolic because the products are slightly more complex possibly compared to the starting molecule, and that's going to utilize or take in some energy. So we could actually say that is endergonic. So during respiration we have a number of different redox reactions where we get reduction and oxidation occurring at the same time. Now we've got molecules called co-enzymes, and there's two main co-enzymes that we find in respiration, NAD and FAD. Now these co-enzymes, they're not actually enzymes themselves, so they're not proteins, but they're molecules that can bind hydrogen. So they act as carriers of hydrogen, or if you like, taxis of hydrogen, and they carry the hydrogen to the electron transport chain that we find at the inner mitochondrial membrane. So inside cells we find NAD excuse me, in its oxidized form as NAD+. So we'll find this inside the cytoplasm and also inside the matrix of the mitochondrion. Now, in reactions that release hydrogen, this hydrogen gets picked up by the NAD coenzyme. So we know it becomes reduced. And that's often written as NADH/H+. So really what's happening here is the NAD, the oxidized version, actually binds with 2 high energy electrons and 1 proton. And that actually forms the reduced NAD. Now the other proton also is available, and that's called reduced NAD. Now the other coenzyme that we need to know about is FAD, and this one's only involved in the Kreb cycle that we'll do shortly. So FAD inside the matrix of the mitochondrion combines with 2 high energy electrons as well as 2 protons to form reduced FAD. And again, that carries the hydrogen that contains the 2 high energy electrons to the electron transport chain. Now, how do the coenzymes work? Well, if we've got an enzyme such as this structure here, number 1 in orange, the substrate in this case is called malate. And that's a 4-carbon substrate that we actually find in the Kreb cycle that we'll see later. Now that binds to part of the active site because it has a complementary shape to this particular active site. The other substrate in this case is oxidized. So we can put the oxidized NAD. So this is the coenzyme because it's needed for this particular enzyme to perform its catalytic activity to produce product. So another part of the active site of the enzyme is complementary in shape to the shape of the coenzyme oxidized NAD. So both malate and oxidized NAD bind to their respective parts of the active site. The enzyme then catalyzes the removal of hydrogen from malate, and that hydrogen gets transferred over onto NAD. So malate loses hydrogen, so that is oxidized. So we can say here malate is oxidized because it loses hydrogen. The oxidized NAD gains hydrogen, like we can see here. So NAD becomes reduced NAD. So because we've got an enzyme reaction, we've got an intermediate in respiration called malate. That becomes oxidized. The coenzyme called NAD becomes reduced. So because we've got oxidation and reduction together, we call this a redox reaction. So it's redox, and that means reduction and oxidation occurs side by side in the same enzyme active site. Now, because the products are a slightly different molecular shape compared to the substrates, the products are then released from the active site. Now, malate was the original substrate, the 4-carbon compound that we get in the Kreb cycle. And the product that gets released is actually called oxaloacetate, which is the next intermediate, the 4-carbon intermediate in the Kreb cycle. So malate becomes oxidized, oxaloacetate is formed, and that's released from the active site. NAD becomes reduced, and that is released as reduced NAD. And the NAD will carry the hydrogen to the electron transport chain in the inner mitochondrial membrane. So this is one example of a redox reaction that we find in respiration. Now, aerobic respiration, we're going to see, occurs in 4 main stages. The first stage is called glycolysis, and this is where glucose is originally broken down. Now, glycolysis only occurs in the cytoplasm of cells. So this has nothing to do with the mitochondria. this will actually produce a net, we'll see in a minute, of 2 ATP by what we call substrate level phosphorylation. So glycolysis on its own does produce a small amount of ATP, but it will also produce some intermediates that move into the mitochondria that then enter the link reaction. So this occurs in the matrix of the mitochondrion. Now the link reaction makes a product called acetyl coenzyme A, and that delivers a 2-carbon component into the Krebs cycle, which also occurs in the matrix of the mitochondrion. Now the Krebs cycle is very useful because it actually produces 3 reduced NAD and 1 reduced FAD. So it's a good source of reduced coenzymes. Now these coenzymes, when they're reduced, they will move to the electron transport chain. Now this occurs in the inner mitochondrial membrane, and it will use the high energy electrons from the reduced coenzymes to power some proton pumps that will build what we call an electrochemical gradient. So that electron transport chain is directly linked to what we call oxidative phosphorylation that makes a high level of ATP molecules. So we're going to start with glycolysis. Now we said this occurs in the cytoplasm. It does not require oxygen, and it can occur under anaerobic or aerobic conditions. So anaerobic conditions, we will have a lack of oxygen, so lack of oxygen. And we know aerobic conditions will be in the presence of oxygen. So we get glycolysis under both of these conditions. So this is the first step that occurs in the cytoplasm, and all the enzymes involved in glycolysis are actually coded for by genes inside the nucleus of the cell. So the glucose, once that enters the cell, normally by facilitated diffusion or possibly active transport, it immediately becomes phosphorylated, and that forms a hexose sugar with a phosphate group on the end called hexose phosphate. So that's going to utilize 1 ATP because the phosphate group is transferred directly onto glucose to form hexose phosphate. The hexose phosphate is then phosphorylated a second time using another ATP. So we end up with hexose bisphosphate. Now this 6 carbon with the 2 phosphates attached is actually highly unstable. So one of the reasons why glucose is phosphorylated is to make the unstable 6 carbon hexose bisphosphate. Now what happens to that is, because it is unstable, it splits, and what we end up with is a molecule of triose phosphate, but because that's a 3 carbon, we're actually going to have 2 of them. So the unstable 6 carbon with 2 phosphates on it splits in the middle. We have 2 triose phosphates that are formed. Now each triose phosphate, if we start with this one on the left, will undergo 2 types of reaction. The first of which is the loss of hydrogen, and that's by an enzyme called dehydrogenase. Here at the bottom, dehydrogenase. So the triose phosphate is oxidized through the loss of hydrogen, and the hydrogen gets converted and transferred onto NAD to form reduced NAD. Now the second type of reaction is the loss of the phosphate group, and that phosphate group is directly transferred onto a single ADP, and that will form 1 ATP molecule. Now an inorganic phosphate in the cytoplasm is then also transferred onto ADP, a second ADP, using the energy from this reaction. So we end up with triose phosphate being converted to a 3 carbon pyruvate with the production of 2 ATP and a single reduced NAD. Now that is per triose phosphate. So if we look at both triose phosphates, what we end up with is 4 ATP being produced by substrate level phosphorylation, and a second, in this case, reduced NAD being produced by the activity of triose phosphate dehydrogenase. But if we look at the initial step at the top, we can see it's called the investment phase. And we know 2 ATP are needed to phosphorylate the glucose to produce hexose bisphosphate. So the net production of ATP here is not 4, but it's 2 ATP, because 2 of the 4 are used in the investment phase to phosphorylate glucose twice. Now we will have 2 reduced NAD also being produced from glycolysis. Now the other reason that the glucose is phosphorylated is because in the cell membrane, we draw a phospholipid bilayer at the top here. There actually isn't any carrier proteins for hexose bisphosphates or hexose phosphate. So these are the phospholipids and the phospholipid bilayer. Now we know the carrier protein here is an intrinsic protein, and we know glucose can enter the cell through this. But if there's a lack of a carrier protein for hexose phosphate or hexose bisphosphate, it means the hexose phosphate cannot move out of the cell, and so it enters glycolysis, where it gets converted eventually to pyruvate. So it kind of traps the glucose inside the cell, and then it gets used directly in glycolysis before it can diffuse out of the cell. So we can now summarize glycolysis, and we know if we look at the numbers of reduced NAD molecules being produced from glycolysis per glucose, we're going to get 2. Now we know the reduced NAD carries the 2 high energy electrons to the electron transport chain, and those electrons will be used eventually in oxidative phosphorylation. But also, just in glycolysis per glucose molecule, we actually get 4 ATP being produced by substrate level phosphorylation. But 2 of those are invested into the phosphorylation of glucose to form hexose bisphosphate in the first couple of steps of glycolysis. So the net ATP per glucose per glycolysis is 2. Now, once the pyruvate is formed in the cytoplasm, there are carrier proteins in both the outer and the inner mitochondrial membrane for pyruvate. And these allow the movement of pyruvate into the matrix of the mitochondria. So the matrix. Now we know pyruvate is a 3-carbon organic molecule. Now 2 reactions happen to pyruvate in the matrix. There's an enzyme called pyruvate dehydrogenase, and we know that removes hydrogen from the pyruvate, and it adds the hydrogen onto oxidized NAD. So our oxidized NAD becomes reduced, and we know the reduced NAD is going to be carried to the inner mitochondrial membrane, and that's going to be used in oxidative phosphorylation. So we know NAD is a carrier of hydrogen and all that energy in the electron or electrons from that hydrogen. Now, not only is pyruvic acid or pyruvate oxidized, which results in reduced NAD, but it's also decarboxylated. So the 3-carbon has a carbon removed as carbon dioxide. So this is the first carbon dioxide molecule produced of the 6 per glucose. And that leaves a 2-carbon compound called acetate. And the acetate combines with coenzyme A to form acetyl coenzyme A. And the job of the coenzyme A is to carry the 2-carbon acetate into the next stage, which is called the Kreb cycle. So to summarize, pyruvic acid or pyruvate enters the matrix of the mitochondrion through carrier proteins. We know it's oxidized through dehydrogenase enzymes, so we get reduced NAD. We know it's decarboxylated to form a 2-carbon fragment called acetate. That acetate joins with coenzyme A to form acetyl coenzyme A. That carries the 2-carbon acetate into the Kreb cycle. Now, the Kreb cycle then, it's a series of chemical reactions in the matrix of the mitochondrion. Now, all the enzymes involved in the Kreb cycle are coded for by genes that we find on the small circular piece of DNA inside the matrix of the mitochondrion. Now, there's a lot of chemical energy still locked up in the acetate group combined with coenzyme A called acetyl coenzyme A. So that coenzyme A delivers the acetate group directly to this series of chemical reactions. And the products here are going to be 3 reduced NAD, 1 reduced FAD, and also 1 ATP made directly by substrate level phosphorylation. So we'll look at each step in a little bit of detail. So the starting molecule is going to be a 4-carbon compound called oxaloacetate. And this is an organic molecule in the matrix of the mitochondrion. Now this gets combined with the 2-carbon acetate fragment carried into the Kreb cycle by coenzyme A. So if you add a 2-carbon fragment acetate with the 4-carbon oxaloacetate, we get a 6-carbon compound that we call citric acid or citrate. So we can see here on the left we've got a 4-carbon molecule, we've got a 2-carbon acetate, and we end up with a 6-carbon molecule called citrate. Now the citric acid or citrate is essentially oxidized, and it's also decarboxylated. So if you take a 6-carbon citric acid molecule, it's oxidized, so it loses hydrogen, and that hydrogen gets added to NAD+, which is the coenzyme, so we end up with reduced NAD. We know a molecule of carbon is lost as carbon dioxide, and that would be citrate decarboxylase, and we're going to end up with a 5-carbon compound called alpha ketoglutarate. Now the alpha ketoglutarate, the 5-carbon organic molecule, undergoes several different reactions. We know it's oxidized, so it loses hydrogen. So here we get a second reduced NAD in the Kreb cycle. We know it's also decarboxylated, so the 5-carbon compound will lose a carbon as carbon dioxide down here at the bottom. And we get another reaction that occurs called substrate level phosphorylation, where a phosphate group, inorganic phosphate, is combined with ADP to make an ATP molecule directly by, as we've said, substrate level phosphorylation. Now, the 5-carbon alpha ketoglutarate, we know it loses the carbon as CO2, we know it loses hydrogen, so it's oxidized to form reduced NAD, and we know ATP, one of them is made directly. That leaves a 4-carbon compound called succinate. Now, succinate can be converted by a series of reactions to malate that we saw earlier. And that's going to lose, the succinate is going to lose hydrogen, so it's oxidized. But this time the hydrogen gets picked up by the coenzyme called FAD. So we end up with reduced FAD from this succinate to malate reaction. Now the malate is converted by a series of reactions to oxaloacetate, and it essentially loses hydrogen, so it's oxidized into oxaloacetate. And again, this hydrogen is picked up by the coenzyme NAD, so we get a third reduced NAD molecule. So from the Krebs cycle, we have 1, 2, 3 reduced NAD. We have a single reduced FAD, and we have 1, 2 carbon dioxide molecules being formed. Now, because the Krebs cycle goes around twice per glucose, essentially 4 carbon dioxide molecules are being released per glucose in this series of reactions. Now, we know a single carbon dioxide is lost in the link reaction, but because 2 link reactions occur per glucose, the link reaction will lose 2 carbon dioxides by decarboxylation. So that's where the 6 carbon dioxides come from in the original starting equation for aerobic respiration. 2 CO2 from the link reaction and 4 CO2 from the Krebs cycles, which we get 2 of them per glucose. In some textbooks, you might see the Krebs cycle drawn in a little bit more detail. So there are a number of different steps that you could add in that we didn't show on the previous slide. So for example, citrate is converted to isocitrate by an isomerase enzyme. And it's the isocitrate that is decarboxylated and oxidized to form the 5-carbon alpha ketoglutarate. Now, the alpha ketoglutarate we know is also decarboxylated. We know it's oxidized, so it loses hydrogen, which is reduced NAD being produced here to form succinyl coenzyme A, because it also involves the addition of coenzyme A at this point. Now that loses the coenzyme A, and that will form succinate. We know ATP is formed during this step by substrate level phosphorylation. Succinate is converted to fumarate, which is an oxidation reaction, which results in the reduced FAD. Fumarate is then converted to malate, the 4-carbon organic acid, and the malate is oxidized to oxaloacetate, resulting in reduced NAD being produced. So this is a little bit more of an extended Krebs cycle, if you like, in a little bit more detail. So if we summarize the Krebs cycle, then we can look at the number of products being released. Now, we know per cycle we're going to get 3 reduced NAD being produced, but we know the Krebs cycle goes around twice per glucose, because in glycolysis we actually end up with 2 times pyruvate from glycolysis, and we know each of these pyruvate enters the link reaction to form acetyl coenzyme A. So 2 times pyruvate will produce 2 times acetyl coenzyme A from the link reaction, and each of these goes into the Krebs cycle, so we get 2 cycles per glucose. So actually, in summary, we don't get 3 reduced NAD per glucose, we get 6 reduced NAD per glucose, because the Krebs cycle goes round twice. So we get 3 reduced NAD per cycle, per Krebs cycle, and that goes around twice per glucose, so we get 6. Now we know from each Krebs cycle we get 2 CO2 from the 6-carbon citric acid to the 5-carbon alpha ketoglutarate, and from the 5-carbon alpha ketoglutarate to the 4-carbon succinate. But because the cycle again goes around twice, we're going to have 4 carbon dioxide produced here. Now we know from the link reaction we get 1 CO2 being released by decarboxylase enzymes, but because we get 2 link reactions per glucose, we get 2 carbon dioxides from the link reaction by decarboxylation. So 4 plus 2 is 6 carbon dioxides, which matches our original starting equation. Right, we know from each Krebs cycle we get 1 reduced FAD, but because the cycle goes around twice per glucose, we end up with 2 reduced FAD per glucose. And again, we know we get 1 ATP by substrate level phosphorylation, where inorganic phosphate is added to ADP using the energy from the alpha ketoglutarate to succinate reaction. So ATP is formed directly by substrate level phosphorylation, but the cycle goes around twice, so we end up with two ATP per glucose from substrate level phosphorylation from the Kreb cycle. So per glucose, we get 6 reduced NAD from the Kreb cycle. We get 4 carbon dioxide. We get 2 reduced FAD, and we get 2 ATP being made directly. So what is the role of the reduced NAD and the reduced FAD? Well, we know they're reduced coenzymes, and we know they carry hydrogen to the inner mitochondrial membrane. And here we get a series of proteins embedded in the inner mitochondrial membrane called the electron transport chain. Now, if we look at NAD, or reduced NAD, that actually is NAD plus with what we call a hydride ion and a proton. Now, the hydride ion, so 2H minus, is actually made from a proton and 2 electrons. So really, reduced NAD carries a proton and, more importantly, 2 high energy electrons. So reduced NAD carries 2 high energy electrons to the electron transport chain. Now, if we have a look at this electron transport chain in a little bit more detail, we can see on the left here we've got reduced NAD. We know it carries 2 high energy electrons to the ETC as well as protons. Now, at the first proton pump, the reduced NAD becomes oxidized back to its oxidized form, NAD+. So we know it loses hydrogen ions and also the 2 high energy electrons. Now, when these 2 high energy electrons move across proton pump number 1, they lose a little bit of their energy, and that activates the proton pump to pump the 2 hydrogen ions against the concentration gradient into the intermembrane space. So this yellow proton pump is activated by some of the energy from the 2 high energy electrons, and it pumps 2 protons across the membrane into the intermembrane space. So this portion here at the bottom of the slide, this will be the mitochondrial matrix. So we're going to get 2 protons being pumped into the intermembrane space. Now, if you look at the top of the slide, we can see this shaded area. This is the intermembrane space here. So we're going to get protons starting to accumulate in the intermembrane space between the outer and the inner mitochondrial membranes. Now, the 2 high energy electrons that have lost a little bit of energy, they get passed to an electron carrier, and that carries these electrons within the phospholipid bilayer to the second proton pump in green. And as the 2 electrons move through the proton pump, they lose some more energy, and that activates the proton pump to change shape and pump 2 more hydrogen ions into the intermembrane space. Now, the 2 electrons are picked up by a second electron carrier in the diagram here called cytochrome C, and that carries the 2 electrons to the third proton pump, which is actually called cytochrome C oxidase. And the third proton pump pulls the electrons from the electron carrier number 2, and they pass through the proton pump. They lose the last of their energy, and that allows activation of the third proton pump to pump hydrogen ions against the concentration gradient to continue to build the hydrogen ion concentration in the intermembrane space. So the role of reduced NAD is to deliver 2 high energy electrons to the electron transport chain. As the electrons pass down the electron transport chain, they lose energy. It activates sequentially the 3 proton pumps. They become activated, they pump protons into the intermembrane space, and that builds the electrochemical gradient. Now this is important because the electrochemical gradient is going to be coupled to the production of ATP. Now if we look at the diagram above in blue, what we can see is projecting out from the inner mitochondrial membrane towards the matrix are these stalked particles. So you'll always see the stalked particle face out towards the matrix from the inner membrane. And the electrochemical gradient, the protons, the H+ ions, are going to move through the ATP synthase, which is the stalked particle. And the energy from the electrochemical gradient is going to be converted into the high energy bond that we find in ATP. So if we look at the stalked particle in a bit more detail, we know it points out towards the matrix. So behind the stalked particle here, we have an electrochemical gradient that we've just established because of the electron transport chain. Now, protons move through the first part of the stalked particle, and there's a channel for protons to move through the membrane. Now we know phospholipid bilayers, because of the fatty acid tails, they are hydrophobic in the core of the phospholipid bilayer. So we know protons can't just simply diffuse across the inner membrane back into the matrix. So they have to go through the stalked particle. Now when the protons move through the proton channel, it allows this part of the stalked particle to rotate. So we get conversion of potential energy in the electrochemical gradient into mechanical energy. And as this part of the stalked particle rotates, it allows the catalytic domain, so this part in blue at the bottom, that's the catalytic domain of the stalked particle, part of ATP synthase. It converts the mechanical energy back into chemical energy by making ATP from ADP and inorganic phosphate. So the protons move down their concentration gradient through the stalked particle back into the matrix. The potential energy in the electrochemical gradient is converted into essentially chemical energy again in the form of ATP. Now, we call it oxidative phosphorylation because the reduced NAD is oxidized back to NAD+. And that can then go back to the Kreb cycle or the link reaction or back into the cytoplasm for glycolysis to be reduced once again. But the oxidation of reduced NAD allows the establishment of the electrochemical gradient. Through chemiosmosis, the electrochemical gradient is converted back into chemical energy in the form of ATP. Now, what pulls the electrons through the electron transport chain? Well, that's the job of molecular oxygen. So we call oxygen the final electron acceptor, and that's needed to pull the 2 electrons through the ETC. They combine with oxygen as well as 2 protons, and we end up with a molecule of water, which we know is a waste product of aerobic respiration. So we know oxygen is very electronegative. It wants to attract electrons to it. We call it the final electron acceptor. Now, in some exams, you'll be given some drugs that can interrupt oxidative phosphorylation. Now, one of these drugs we know is called cyanide, and cyanide blocks the movement of electrons at this point here. So the second electron carrier, called cytochrome C, is blocked between the third proton pump, so the electrons can't move through this point. So if the electrons can't move down the ETC, it means that the electron flow is interrupted. We know no more protons get pumped into the intermembrane space, the electrochemical gradient gets diminished, and less ATP is made. Any reduced NAD or reduced FAD cannot be reoxidized, and so it has a knock-on effect to things like the Kreb cycle and the link reaction and glycolysis. So this is a summary of what we've just said. It's called oxidative phosphorylation. We know reduced NAD becomes oxidized. It releases protons as well as 2 high energy electrons. They pass down the ETC, they lose energy. That energy is used to activate proton pumps 1, 2 and 3. We establish an electrochemical gradient. The protons move back down their concentration gradient. The potential energy is converted back to chemical energy in the form of ATP, and we pull the electrons through the ETC because of the presence of molecular oxygen, and that combines with protons to form water. Now, if we were to talk about reduced FAD, that delivers the 2 high energy electrons not to proton pump number 1, but essentially to proton pump number 2. So the 2 high energy electrons would move and enter the ETC at this point here. So actually there's going to be less protons being pumped into the intermembrane space as a consequence of the oxidization of reduced FAD. So what we say is for each reduced NAD, 3 ATP are made, because each of the 3 proton pumps are used to pump hydrogen ions into the intermembrane space. But for each reduced FAD, only 2 ATP are made, because FAD or reduced FAD delivers the high energy electrons to proton pump 2. So only proton pump 2 and 3 are utilized in respect to reduced FAD. So if we summarize the electron transport chain and oxidative phosphorylation, what can we say? Well, we know each reduced FAD produces 2 ATP and each reduced NAD produces 3 ATP. And we know that intermembrane space becomes much more acidic. It has a low pH, an acidic pH, because of the accumulation of protons, and the electrochemical gradient becomes established, which generates a proton motive force. So we might get asked a question about the energy budgets for aerobic respiration. So we can summarize this in a single table. So if we start by looking at carbon dioxide then and where that comes from, well, in glycolysis there's no decarboxylase enzymes, so no carbon dioxide is released here. So if we look at lactic acid formation from anaerobic respiration in, for example, skeletal muscle, no carbon dioxide is released from lactic acid formation. However, if the pyruvate enters the matrix of the mitochondrion, we know from a single link reaction 1 carbon dioxide is lost through decarboxylase. So per glucose we get 2 link reactions because there's 2 pyruvates and therefore 2 carbon dioxides. Now from the Kreb cycle we get 2 decarboxylase enzymes that release 2 molecules of carbon dioxide. But we know the Kreb cycle goes around twice per glucose, so we get 4 CO2 from a single glucose molecule per Kreb cycle. Now reduced NAD, well, from glycolysis we have 2 of them being produced. And we know these NAD, when they're reduced, will carry 2 high energy electrons to the ETC, and they will actually be involved in oxidative phosphorylation. From the link reaction we get a single reduced NAD, but because there's 2 link reactions per glucose we get 2 reduced NAD. And likewise from the Kreb cycle it goes around twice per glucose, So 3 reduced NAD times 2 is 6. There's no reduced FAD from glycolysis or the link reaction, but there's a single reduced FAD from the Kreb cycle, so we get 2 FAD reduced per glucose. Now, substrate level phosphorylation. Well, from glycolysis, we get a net of 2 ATP being made directly, where the phosphate group is transferred from a donor molecule onto ADP to make ATP. From the link reaction, there's no ATP being made directly. From the Kreb cycle, there's 1 by substrate level phosphorylation, so we get 2 per glucose because the cycle goes around twice. Now, net ATP by oxidative phosphorylation. Well, that's going to use the reduced NAD and the reduced FAD. So from glycolysis, because we have 2 reduced NAD, we know a single reduced NAD makes 3 ATP at the electron transport chain and oxidative phosphorylation. So here we're going to get 6 ATP because we've got 2 reduced NAD from glycolysis, each of which makes 3 ATP. Now, if we look at the link reaction, again, we've got 2 reduced NAD, each of which will make 3 ATP at the electron transport chain and the ATP synthase in oxidative phosphorylation. So we get 6 ATP by oxidative phosphorylation. Now, this is quite a big step. So from the Kreb cycle per glucose, we get 6 reduced NAD. Now, we know each NAD, when it's reduced, will make 3 ATP from oxidative phosphorylation. So 6 times 3 gives us 18 ATP by oxidative phosphorylation. And we know there's 2 reduced FAD per Kreb cycle. And we know each reduced FAD makes 2 ATP from oxidative phosphorylation. So 2 reduced FAD will give us 4 ATP by oxidative phosphorylation. So 18 plus 4 gives us 22. So now we can add up all these values, and we can see there's 34 ATP produced by oxidative phosphorylation. We can see there's 4 ATP produced by substrate level phosphorylation, and that's going to give us a grand total of 38 theoretical ATP from a single glucose molecule. Now, the actual net ATP is normally less than 38, so it's anywhere between 30 to 32 ATP. And that's because some of the potential energy in the electron transport chain is actually lost as heat energy. So we know respiration, aerobic respiration inside cells releases some heat energy, and that's used, carried around the body in the blood to maintain the core body temperature in mammals. So some of the energy of the electrochemical gradient is lost as heat. We know some ATP is needed to allow pyruvate to enter the matrix through carrier proteins. So that's further going to diminish the theoretical ATP being produced. And actually some of the intermediates in the Kreb cycle, like alpha ketoglutarate, can often be used in other biochemical reactions, for example, amino acid metabolism. So that might also affect the theoretical ATP and bring it down towards 30 to 32 as opposed to 38.